Where we were last time is we were talking about pneumonia, and we were looking at this particular gene here called the Urn B gene, which is a gene which comes from the well, it's been found in the Streptococcus pneumonia, but this gene has been found to be changed so that it has now resistance for any kind of it's showing that this type of pneumonia, I'll just read this, is now actually that's not, but not, it's supposed to be now believed to have always been resistant, or not to be resistant to drugs, but now it is resistant to drugs. And they're thinking that because of this resistance, it could have been horizontal gene transfer. And so we use BLAST just to look around to see whether, for instance, are there any pieces of this gene which are also common in some other organism, which might suggest that that common piece is the piece that was somehow that gave the genetic code for the resistance. Something happened to this gene so that now it's resistant to to different types of therapies that you'd use to get rid of pneumonia. And so we're trying to check to see whether there's some kind of if that gene had any trouble there at all. And so that's really what all this was about. And so then we used two different methods. I'm just kind of flying through these slides here, but I hope you guys kind of remember some of this stuff. It's actually quite good, and you have all this in your slides, and so I hope that you can you can go back again, and when you run new analyses here, you can kind of revitalize your memory from looking at these slides. But we pulled over the file as a FASTA file, and we did that because we were thinking that perhaps we'll go back to this file again and run some more analyses. And so it would be easier for our pipeline if we just had a file someplace in our computer that was just this Streptococcus record, and in that record we wanted to have this FASTA, the genome. And the FASTA format was not a file like this, but rather it just has the name, as you see at the top here, the Streptococcus algecticae strain KMP401. And so on and so forth. That would be the name, and then at the very end, or then the other line, in the other line of the FASTA file, you'd find the sequence, and the sequence would, I don't have a sequence here, but it is there. And so that's what we collected, just this two-line file called the FASTA file, with the name and the sequence. And then we used that for, we went to the sequence in the file and just copied it out, and this is what we got. And this is our sequence. And then we plugged that sequence into BLAST like this, using the default settings, and a bunch of other sequences came up. And so this is kind of like where we left off last time. And what we see here is that these are sequences which have some common element with the sequence that we were looking at previously. So in one of these sequences here, we could make a point or make an argument that the resistance to therapeutics came from one of these organisms. One of these organisms had to have been in physical contact with our original organism that we're looking at, and transferred over this Urn B gene, which was the one that was, which was the gene that has some kind of code for resistance. It's kind of like this resistance in some ways is kind of like tracking viruses here, because viruses continually evolve and they make themselves stronger and smarter, and sometimes the therapeutics that we have to overcome viruses stops working because some kind of natural evolution. If you think about it, the virus is being pressured by evolutionary pressures to survive. And so anything that they can find that would help it to survive is going to be a good thing. And so that's going to be something that's going to stick. It's going to be a conserved. It will stay in its genome because it helps the organism survive. And so this gene that we found, which is similar to any of these genes here, which provides some kind of a resistance to therapeutics, which would stop pneumonia, is the same kind of case here. If the gene dies because we're able to stop pneumonia or stop this this coccus or Streptococcus virus, or not virus, bacteria. If we're able to stop that bacteria, then that means that the gene will itself go away. But in this case, this gene appears to provide some evolutionary strength to this, or evolutionary advantage to this organism, because it's able to survive in spite of our therapeutics. So we're thinking that one of these organisms here may have actually been the one that got close enough to our first organism to transfer over some kind of piece of genetic code which gave it this resistance. Now we looked at the scores, and in our results, we have these big bars of red. These bars of red, I believe, are the common areas. So anytime you see, in fact, you don't see the names of these things, but each bar of red that you see here actually stands for its own sequence from our list. So it's one of these. So you can click on these things, and the idea now is that when you have sequences of difference, where you have like different regions, and maybe the alignment isn't absolutely perfect, then your red line is going to have like kind of holes in it. You'll see kind of like where holes exist and where red patches also exist, and that will allow your eye, as you're looking down the page here, to see where those common regions may be. But this is saying that these sequences that I found are almost, well, I mean, they're exactly the same. I don't see any breaks in these lines at all, and so that suggests to me that the sequences are absolutely the same. And another point that we made last time was that because these sequences are so similar, because they are the same gene, I would wager that this gene is actually a very important gene, and that is that it does something that's essential to the survival of the organism. Because if anything happened to that gene, and it could, we've studied mutations, or we've studied, other kinds of ways that genes could somehow become different: insertions, mutations, deletions, substitutions, horizontal gene transfer, all of those things, even viruses, for instance, that bring information from one organism to another. If they corrupt this gene, maybe the gene doesn't function, and maybe that functionality will cost the organism its life. And so therefore the broken gene is going to be phased out. It's not going to stay in the gene pool. But because there's so much similarity here, this suggests that this gene is actually quite important. And it is, because this is the gene that provides the immunity to therapeutics for pneumonia. So it makes sense. What I'm saying is the results should make sense. And you, as the bioinformatician, as you look at your results, you need to decide for yourself, does this make sense? Do you buy this? Does it seem believable? Because you'll get results, and then you'll get results, and the results some of the results you'll look at will be, yeah, that's just random. It can't possibly be true. But other results you'll look at and you'll say, actually, that explains a theory that I have, and that is that if this gene were to be broken, then the organism dies. And so you have to think in terms of what's the advantage and disadvantage of having that gene working. These are just more results showing how identical these particular genes are. And so we conclude that because there was a 95% similarity between these genes, and here we found this is 100% similarity, so I guess that's even better. But because it's so similar, we could conclude that it was horizontal gene transfer. And we could make a hypothesis that it was. The next step, we have to do more research here, but the next step is to determine, were these organisms actually closely, in contact with each other? Did they actually have anything? Were they together in some way? Okay, so that's actually quite exciting. This is the kind of analysis that really blows my hair back, and I'm hoping that you're feeling it too, because this is just, it's like you're investigating the I guess a biological crime, if you will. Some gene is cheating, and it's somehow able to overcome evolution, and so you're finding out like what that thing was. But now, because we were working with DNA. As you know, proteins can also be represented as strings. That means you can actually take a piece of protein, and you can put out, you can use a character that represents each amino acid and spell out a long string which represents that particular protein. And again, just as we saw before, we can create trees and other kinds of evolutionary study mechanisms to determine how one protein is related to another. And so that's really what we're talking about today. How is one protein related to another protein? And I think you'll find that this is quite exciting. And so you can, again, we're going to use the same types of skills that we've learned before, which is why we've spent so much time doing alignment, because alignment is absolutely one of the central pieces of analysis in bioinformatics. I mean, it seems like all studies, in my experience, it seems like all studies with DNA or gene study, they all begin with alignment. Then they go into other places, but they all begin with alignment, and those alignment scores give you trees like this, so that you have a starting point, so you know how closely related one thing is to another thing. Anyway, so let's move on now. And so you can take proteins, and you can also blast them in blasts, as we have seen before. I mean, we were working with BLAST, and so you can, I mean, that was working with, that was used to look at DNA, and so you can actually dial up a protein, if you will, like the record of a protein in NCBI, and pull up some information about proteins and see how they are different. But one thing about proteins, though, is that you'll see that there's this word predicted. You'll see that sometimes proteins have to be predicted. And so this is where things get a little bit, things get interesting here. And that is that when you're working with DNA, you can just say, I have the DNA sequence, I know this is the sequence, and therefore I can compare this sequence to another sequence, which I also know. But in proteins, you don't necessarily know that that's what the sequence really is. And the reason for that is because sometimes you have a piece of protein and you're going, and you don't really know how, I mean, what I'm saying, you don't know what the actual DNA was that gave you that protein. And we're going to talk about that today. But just remember, though, that sometimes you don't know. I mean, if you think about it, actually, when we looked at the codon table, there's different ways of actually writing out, or I guess translating each codon into an amino acid. Each amino acid has multiple triplet codes from DNA that you can use to actually spell out or translate that amino acid. And so therefore, if you're working with a protein, sometimes when you do some kinds of, or certain forms of analysis, you have to go back to the DNA and then figure out where that DNA came from, and then to figure out from that DNA what proteins could I have, and then compare those proteins. It's we'll talk about that in about 10 minutes. But it's very cool. But so you have this predicted thing here. So you don't know, looking at a piece of protein, what exact DNA you use to get there. It's just it's a non one-to-one function, as we'll see in just a moment here. So this takes us into this kind of discussion here, where, for instance, look at this is your central dogma of biology. And you can see that on the very top here, the 5', 3', don't forget that. That's necessarily your DNA. When you have the two pairs together like that, you have the 5', which begins with ATG, and then so forth and so on and so forth. And then underneath that, you have the 3', so that's your helix. Then you have your transcription, which gives you the RNA, and then RNA gives you translation that gives you the protein. So you can see that each of these proteins, you have, typically your proteins will begin with methionine, and so that's your MET that begins on your protein on the bottom line. That is your beginning of your protein. But the thing is, though, that we have seen that sometimes you don't have, sometimes it's not so straightforward as how that protein was actually created. Methionine, there's only one triplet code that gives you that, and that's AUG from RNA. But you have other things like lysine, for instance, and serine. Like serine is the second one here, which UCU from the RNA code could have given you serine, but other stuff could have also given you serine. And so you can see now, if you're starting with protein and you're working your way back, you don't necessarily know what the code was exactly that gave you that protein. So we're going to talk about mutations now. And so that's kind of like where the mutations idea comes from. And that is that you have different ways of spelling out these amino acids that make up your protein. And you could get some kind of a mutation that occurs in your DNA. But by looking at the mutation, or by looking at the protein that comes out of that DNA that's transcribed into RNA, then translated into protein, you still get the same protein. And so there's really nothing different. And so if you're looking at that protein, you'd never know that there was any kind of trouble that happened with that, with the original sequence of DNA. In this case, for instance, at the DNA level here, where it says lysine, the TTT, that T that's in red there, is what they call the silent mutation, because there's other ways of spelling out lysine. TTT, and I think that even TTC, in fact, we'll see that just in a moment what it is. But you wouldn't know by looking at the protein that there was any problem. So that's why they call this a silent mutation, because yes, there was a mutation that happened, but it doesn't mean that the mutation was enough to change the protein. Now, let me just say something else. I'll let you in on a little secret, and that is that I keep talking about changing the protein. What does it matter if the protein gets changed? Like, who cares? Well, actually, the protein cares, because supposing now you're all in your rooms right now, I'm thinking, or some of you may be outside, but I'm talking to the people who are inside their dorm rooms. Imagine for some reason that the lock on your door became different. and suddenly the key that you used to open that lock doesn't work anymore. And suddenly you can't open the door, and you can't leave the room, and you can't leave the building, and you can't continue your life because you're locked in your room. And so if you change the function of how that lock works, then you change the function of how you can actually leave the room, and a bunch of other catastrophic events will occur too. And so by changing your proteins in some way here, you're going to actually change its function, and a bunch of other catastrophic events are going to happen as a result of that change. I mean, and you can't always tell what those events were going to be. Anyway, so we're talking here about the silent mutations. We've already talked about this a little bit, but these mutations are actually, they're actually quite important though, because there's a paper that comes from Nature journal. Nature is like one of those big it's like one of those big journals that exists in science. And if you get a paper in Nature or Science journal, Nature or Science, Nature is from England, Science is from America, but if your paper shows up in those journals, you have it made. I mean, it's like your research has been accepted by everybody. It is you've become a celebrity. And so here's a paper that's come out of Nature. Silent mutations speak up. Overlooked genetic changes could impact on diseases. And so going back to this, that's saying that even though you get the same protein that comes out, if you have a mutation in your DNA, but you still get the same protein, you could still suffer some risk. And that makes it difficult to know that you have this mutation, because if someone analyzes your protein, they find out the absolute, the composition of the protein. They found it's made out of lysine, lysine stop, arginine, and threonine, or whatever they may be. If you find out that that's what it's made of, then you'll just say, well, it's the protein you're supposed to have. I don't see how this is a bad thing, because the wild type protein is supposed to be this exact combination. But they're not looking necessarily at the DNA level, which is where the mutation happened. And so that's why they call this a silent mutation. So I'm just going to go on a kind of a divergent track for just a moment here. The reason why I've changed your slides from last night is because I kind of came across this database. I've used this before, but I'd forgotten about it for some reason. I just forgot about it. But there's a database out there that allows you to kind of keep track of some of the genes for which silent mutations have been found. I wanted to take you for a second here just to show you, I don't think I can copy, or I don't think I can click on this thing. I have to, here, let me see what I'm going to open up this paper and just kind of show you what this paper is, if I can get this. And hopefully it'll show up. Okay, so there's this database here called the DBDSM. It's kind of hard to say. It doesn't quite. It doesn't. But it's basically a database for the what do they call this? Deleterious synonymous mutations, which is another way of saying silent mutations. But in this case, it means like they're not just silent mutations, but they're mutations that cause damage. But I want to read you something here. For instance, if you look at the abstract of this paper here, the synonymous mutations which change the sequence of a gene without directly altering the amino acid sequence of the encoded protein were thought to have no functional consequences over time. This was a very long-standing take on how silent mutations behaved. Okay, so what? You've got a silent mutation, the protein's still the same, you're fine. It's that kind of thinking here. They were thought to have no functional consequences. They were often assumed to be neutral in models of mutation selection and were completely ignored. And look at this, completely ignored in many studies. That's really dangerous. I look at this thing in with two different ways of understanding it. I think that I'm glad that the science is here, but also that I'm kind of horrified that any kind of data anywhere in science was ignored because it wasn't thought to have anything in there. All data has something in it. You just have to find it. And in this case, if it was just ignored, then that's bad. But here we are. This is the next big line here that kind of delivers the main take. However, accumulating experimental evidence has demonstrated that these mutations exert their impact on gene functions via splicing accuracy, mRNA stability, translation fidelity, protein folding, and expression. And in some of these mutations are implicated in human diseases. So let's just stop and appreciate this for a second. Here's a whole wide there's a whole wide variety of data that's sitting out there, which could explain, for instance, why people are having certain types of health-related issues, and it's been ignored because, you know what, it doesn't seem that important. And so here's a database, I just wanted to show you that part here, but here's a database that's available free online at this, hopefully you can see what I'm looking at now, but it's down here, but it's a database that's available free online, and it keeps track of genes, which you can use to kind of see where, to see which genes have actually been recorded as having silent mutations and which silent mutations were actually, had very serious consequences for those individuals who had those silent mutations. And so this is actually kind of a cool database. I mean, I'm glad that somebody put this together. This must have taken a long time because it would have been curated, which means that some person would have to go through all the literature and decide, this is a study that's talking about a gene. This gene has been shown to be, somehow related to some kind of disease as a result of a silent mutation. And that means that the study would also have to mention that there's a silent mutation. And so a lot of work has gone into each one of the records that comes out of this. And so we don't necessarily have to go through the whole tool, but if you're interested though, you can try this right now. In fact, you just type in cystic fibrosis in the search bar. I have the quick link that you can see. And when you do that, you're going to get these genes that will come up. Here's the results. Which genes have been altered by silent mutations? Maybe I'll just go ahead and try this just because I'm keen. So I'll just go ahead and I'll just copy this because I'm lazy. But I'll go back to this over here. Hopefully you can all see where I am. I'm going to go to my search. I'll type in, for instance, up here. In fact, this is a nice website, by the way. And not that I'm telling you that you have to use this, but I do like the idea that you have here this EG over here. If you go ahead and just click on this stuff here, it actually does add like sample data that you can put in here, so that you can kind of see how the tool works without having to first know some disease or some specific gene to query. Some tools out there will have that. Anyway, I'm just going to go ahead and put the disease in here. I'm going to hit submit and see what comes up. And here we go. These are genes for which silent mutations have been recorded. So there have been, I mean, again, we must respect this data because this data represents suffering, loss of life perhaps, but certainly suffering. This is cystic fibrosis data, so that means that some person had to have exhibited this particular gene in order for them to for this gene to be uncovered. Somebody had this issue. Okay, so you can go back to the slides here, and you can get this page here. But what's interesting though is that you see these genes that are like running up and down the page, and these genes are those genes which have been affected by silent mutations. Someone found that. And so now what you can do is you can click on, at the very, very rightmost, there's this DB DSM ACK number. That is your ascension number, I think it is, or at least it's a primary, so it should be the primary key that NCBI uses to recognize each protein. If you go back over here, I can click on, for instance, this over here, 072. I'll just open this in a new tab, and I get this data here. And this is the data. It's not very much, it's just about 20 lines, I guess, of information. But this information explains so much. It explains why the patient who had cystic fibrosis had cystic fibrosis, where it happened in their gene, where did the problem originate? The problem happened in DNA, and it was found that some cases of DNA, which were wild type, had Gs, and those people who had cystic fibrosis from the same DNA had, in place of G, they had an A at position 489. I mean, if you think about for a second how much effort it would have taken a person here to go through and to find that at position 489 in the code of that gene, a G they found an A in the case of the cystic fibrosis patient where it should have been a G. I mean, there's a lot of computation involved in that, and so that is why this is a computational-based course. So going back to the slides for just a second here, let me just show you something else. These are kind of like what the big pieces are that I've used in the past to see, to kind of understand what these records mean. But for instance, we have information about which gene is actually affected. And this gene is called the CFTR gene. It's a fairly But I mean, you find it, if you did a blast search on it, you'll find that many organisms, many organisms have the same gene, but you'll find that many organisms don't necessarily have A's where G's are, because that represents the substitution. That's one of the problems of cystic fibrosis. And so over here, for instance, this is just more information about like what the silent mutation was. This is a G, which was replaced for an A. And you have this, if you click on this, oops, I don't want to click on it just yet, but if you go back to the slides, if you click on this bar down here, I'll just go ahead and open this in a new tab. But if you click on the last, what do you call it, speaking bubble, you'll actually find the record that I found of this gene at NCBI. If you go and find the nucleotide, which is right down here, you can actually find this gene, which we're looking at, which is called the, what does it say on the screen here? Right here, it's right here, the CFTR gene. So if I go over here, I'll just do a simple search here, CFTR. So you can see that this whole protein is about the CFTR. And so if you go down to the bottom here, you'll find that there's all these different pieces of information about some of the different genes which are embedded, or the different proteins which are embedded in this particular strand of DNA. But you'll find that this part here, this CDS, or this coding region, right down here in the record, is actually describing the particular area of the gene that we're studying. What am I talking about? Well, for instance, let's go back over here. You see on this gene over here, it says at position, I'll zoom in here, at position 489, right there. That, what is that? You know, where does that actually exist? Well, if you go back to the record here, you'll see that right here, this coding region, this is saying between, in DNA, in the DNA code, which is located at the bottom of this file, between position 133 and 4,575, between that area in the code is all the, the coding region for this gene CFTR. And so this is what the protein should look like. And there's some other information there too that you can use. But basically it's giving you some information that this protein, or that particular product of that gene, is supposed to be this. It's a chloride channel controlling ATPase. Anyway, it's just some fairly basic regulatory function that protein does. But we're saying though that inside this area, inside the coding region of 133 to 4,575, inside there, that is where the mutation actually occurs that renders this whole gene that brings out cystic fibrosis. And so if you actually go to the record itself and you look inside that area, in fact, this might be a different, I think this result is the result that we're looking at for this particular thing on the thing. You'll see that in this search, the search that we just did and the search that's on the slides are different searches. But here we're looking at 489, and so you find that 489 in this sequence here, you find it's basically this G in that sequence, and in this gene CFTR, this G that we can see where this talking bubble is pointing at, that is an A for cystic fibrosis patients. And so this is like what you would call your wild type gene, which is like what it should be, that the majority of the population have. But you still have this. And so you can locate all this information just by doing a query at this, oops, where's the website? At this website here. Just by looking at through this information. And it seemingly all diseases have been added, and it's always being updated every, well, it seems like every week. If you look at this, you'll see that the database has something new added to it. Anyway, so I wanted you to be aware of that, though, and so that if you're going after silent mutations or doing anything where you believe that a silent mutation may have had something to do with the ailment that your patient has, or the organism that you're studying has. Let's say you're studying an organism, you know that the protein is somehow failing, and you're looking at the protein itself, and you find that the protein that this organism has is the same protein that another organism has, but there's something different about them because one of them is sick and the other one is not sick. then you could go to a database like this and see whether that protein or that gene for that protein has ever been found to have any problems. And so that would just suggest that this is just awesome. But it's not as awesome as this picture here. Let me just tell you. There's nothing awesome. Look at this. We have Shui on the drums. We have Darth Vader with an electric guitar. We've got Luke Skywalker. I mean, that is awesome. But this is more awesome. Anyway, so I'll move on here. So let me just get back to the whole idea that we're talking about codons, and we're talking about DNA, which is being basically transcribed into RNA, which is then translated into protein. And I was saying that, for instance, there's multiple ways of going from a piece of DNA or to a protein. For example, look at the very, very top here. You have this PHE, this phi, which is at the upper right. That is a particular amino acid, which is a building block of protein, which can be encoded by RNAs UUU or UUC. It doesn't matter. Either one of those is fine. You will still get the same protein regardless. Look at this over here. If you go down to the next box down, the next group down, you have leucine, LEU, which can be encoded by UUA, UUG, and it continues. CUU, CUC, CUA, CUG. It's like there's different ways of spelling out the same amino acid using DNA. And so that's what we call redundancy. So if you start at leucine, for instance, if I say, here you go, here's leucine, how did I get this particular copy of leucine? You'd scratch your head and say, well, you could have used UUA, I guess, to get to this leucine. Or maybe you used UUG, or maybe CUU, or CUC, CUA, or CUG. It's really unknown. There's no way of actually knowing how you got to leucine from the DNA. And so when I give you a piece of protein to analyze, and you have to go back to the DNA to then compare that protein with something else, or compare that DNA with something else, or the gene code of something else, and then create more protein from that particular sequence of DNA, you may find that your proteins are the same, but the DNA that you're comparing is actually quite different. I mean, very different. But it still seems to give you the same protein. And so we are talking about like these, in proteins, when you're making up proteins, you're using something called amino acids. Amino acids look like these, well, these are the chemical compounds you see at the bottom here. This is actually, we see arginine and lysine. Arginine it's on the right-hand side, second box down, that's arginine, and also right-hand side, third box down, that's arginine, and the other one is lysine here, and so lysine is on the right-hand side, third box down, and so it's, I think there's somewhere else too that exists, maybe not. Anyway, but still, so what I'm saying though is that you have molecules though, two different types of molecules, and they are two different types of amino acids, and they are strung together just as bases in DNA to create the stuff that we call protein. And so that is kind of how they work. But the thing is, though, that what happens if you substitute like an arginine with a lysine, or a lysine with an arginine? We ask ourselves, can this substitution cause harm now or later? Could it ever cause harm? And so to do that, we think about some of these changes, and some of the changes are not necessarily dangerous to protein structure. In other words, these two protein structures are actually kind of similar chemically. And so if you substitute one for the other, is it going to change? And the way I learned this is like, for instance, if you have in your pocket a coin, and you believe that coin is a penny, and then you pull out that penny and see that it's a quarter, it's not exactly a problem here, but it's not the same thing though. But you may not know that there's any difference until you actually look at this thing. And so with these proteins, you may not know that there's any problem until you actually study the situation. And then when you study it, you suddenly start realizing, wait a minute, a lot of people are getting sick from this substitution. It's not working, and so we need to be careful to track this data. So here we go. This is something else that's actually quite interesting here. So we have these amino acid substitutions. This is a kind of a mutation, if you will, kind of a mutation. I mean, it's not like the amino acids themselves are necessarily like, substituted out, but the DNA calls for different different amino acids in those protein sequences, and so that's it's still basically a substitution where you have two protein sequences. One is saying L, and the other one is saying I don't know, LLL, or whatever it is. It's like it's the same kind of thing, and maybe chemically you could argue that they're very similar, but actually it's not the same. And so substituting similar amino acids that have like similar types of properties here, chemical properties, may help you to maintain the protein structure. That is, when the protein, and we'll see this later on, but a protein actually folds into a giant knot, and that knot, how it folds, is the function that it's able to deliver. And so the protein is able to fold into a similar kind of knot because the substitution doesn't really affect it that much. But it is still dangerous, though, because the knot still is not made the way that it should normally be made. And so you can substitute similar amino acids in there, and you can still retain that protein structure, and hopefully you can retain the same function. Structure and function are intimately related in proteins. So if you're studying proteomics, for instance, where it's the study of all things protein, you'll find that if you're studying the function, you're also studying the structure. If you're studying the structure, you're studying the function. The very function of your computer in front of you right now, like the reason that you have the screen above the keyboard and not the keyboard above the screen is a function of how you use your computer. If your keyboard were above the screen, then your hands would always get in the way while you're working, and you wouldn't be able to see what's on your screen. And so if you change that configuration, and you could equally work with your computer upside down if you wanted on your laptop, and that's fine. But you would not be able to really do the work that you were doing before. And the same thing goes for your protein. If you change the structure of the protein, then you change its function, necessarily. If you change the way that the position of your computer, you're changing the way that you can use it. And maybe the new way of using it doesn't help you at all. But if you substitute, if you substitute dissimilar amino acids that have different types of chemical properties, then you may be actually really, really damaging even the protein. That means the function that it used to do is no longer there. And so we ask ourselves, how do we know where these silent mutations have come from? We only have to look at our past discussions in this class. We could use alignment. We can say, okay, well, here's a protein that I know is perfectly healthy, and here's a protein which is not perfectly healthy. Where are the differences? Can I see any differences in substitutions? But this is actually something else here. I'm using a new word now. This is new vocabulary, and that word is called domain. Inside a sequence of DNA, we know that we have coding regions, and those coding regions create proteins. Then you have other pieces of DNA that are not coding regions, and they call those the non-coding regions, or the introns. And the extrons inside DNA are those regions that actually they do something. They create protein. But did you know that inside protein, you also have regions that do specific functions? This is true, and this is baffling at the same time. These are regions in protein which tend to evolve by themselves, but they actually do have specific functions. So, for instance, if you have a car, if you've been in a car, if you know anything about cars, you'll know that cars tend to have radios in them. They also tend to have engines, they have headlights, they have wipers, they have heated rear windows, they have fuel tanks, they have bumpers, they have 4 tires on the ground, they have brake lights, they have all these different pieces. A brake light is not an engine is not a headlight, and a headlight is not the radio. These are specific pieces inside your car which handle specific types of functions. So, for instance, the radio handles the music, the drive is handled by the engine, the road is handled by the tires, and so on and so forth. These are kind of like the domains in your car. These are these little areas, or these particular parts of your car that handle very real types of functions. And inside proteins, it's a very similar situation where you have a domain in a protein which handles a specific type of task that protein absolutely must do in order to maintain its survival. So for instance, a certain protein may have to work with the receptors on the cell. Another part of the protein may have to deal with the immune system and tell the immune system, hey, it's cool, I'm one of the VIPs from this body, and so I'm supposed to be here, so, lay off, man, or whatever. But what I'm saying, though, is that you have these parts of the protein that actually do very important functions, and they're all quite different. And so what you do is you look at those protein domains, you line them up in some kind of in this alignment here, and you see, okay, in all these genes that I have here, and all these proteins that I'm creating here, which are the domains which are slightly different? Because I know that the incidence that's going on here, the problem that I'm looking at, is a result of some kind of protein malfunction. And when I think about the protein, what could malfunction in a protein? Surely the area in the protein that does something. And so that would be your domain. That would be the first place you'd start looking, I would say. But then, of course, there's other places too where damage could hide itself. But I would say start by looking at the domain. And if you change the domain, it means you're basically going back to your car and saying, you know what, I'm just going to take this engine right out, switch it backwards, and put it back in, and see whether that helps. And the thing is, though, that it does not help because your engine isn't designed to be reversed in the hood of your car. And so that would actually damage the function of your car. And so that means that if you wanted to find out why your car isn't driving, then just open up the hood and look at your engine, which has been flipped backwards, and maybe that's the reason why it's not working. And so you're looking at the actual areas of your protein that do things, that have some function, that handle something here. So you're actually asking yourself some questions here when you compare your proteins, and that is, how are these proteins different? How much difference is there? Is it just one amino acid, or is it like several amino acids? Is it a whole bunch of amino acids here? How is that difference actually calculated? Where is the difference located? And then you ask yourself, was this difference due to chance? Am I looking at evolution here? You can actually look at evolution. People do that. You might look at a bacterium, and then you find that when you look at the DNA and you compare that DNA to a more recent bacteria that you actually are working with in your lab, you'll find that the code is not the same. You'll find that the DNA is different, and that therefore the protein is different. And so you've just witnessed evolution in action. You've just seen how somehow the code had to change in order to tolerate your petri dish or whatever it may be. So anyway, you ask yourself, was it different? Was this difference that I find between my sequence and my original sequence, the sequence I'm studying and the sequence that I know it to be, is that due to chance? If it's not due to chance, what could it have been? Evolutionary pressure, a mutation of some kind, just damage from radiation, who knows? And then we ask ourselves the most important question here is, okay, I realize now that my protein is different, it's altered. Does that mean, though, that my changing of this protein has somehow changed the way that the protein functions? In other words, imagine your roommate comes in and says, hey, I'll fix your desk. And you're like, oh, that's great, there's a squeak. Can you just fix it? And they're like, yeah, sure. And he takes out a sledgehammer and just hacks the desk to pieces. And he's like, now you can't use it, but there's no squeak. And you're thinking, that's true, there is no squeak, but actually you've changed the way that I can use my desk entirely, and that is I cannot use my desk at all because you've really removed the function. So you're asking yourselves, is this altered protein actually new and different? Does it have a new function, or has the function been actually removed? And so what you do is you're aligning things all over again. And this is your align. In fact, I pulled this out of where did I get? I think I got this from. There's a paper out there that actually has this. But I like this though because it talks about the conservative, non-conservative, and semi-conservative natures of these replacements. And so they looked at human, chimp, mouse, rat, cow. These are protein sequences. You can tell that they're proteins because they're not A's, T's, G's, and C's, but they are, I see K's, A's, S's, P's, and some other stuff. But I have bases in here which are not DNA bases. I have these, and that tells me that these are actually amino acids. But we have the conservative types of mutations here, which are a change in a mutation basically it provides a kind of a similar physiochemical property, and it also provides miraculously you have maybe a similar effect on function, and it's not going to kill the organism. Basically, it means it's not going to kill the organism. So the conservative change is, yes, there was a mutation. But no, it doesn't really damage the thing so much. And so getting back to the car, you can say that you get a flat tire, you change out the punctured tire with a spare tire in the trunk of your car. That is your mutation, if you will, to your car, and it's a conservative mutation because the spare tire you can still drive on it. The car still functions. It's not quite the same, but it still functions. The tire still turns on your car. You can still get to work. And the idea is that that's how spare tire should be, so that you can still drive to the store and get a new tire or have your tire fixed. Have a tire fixed. It's more green. Then we have these other mutations here, which are called semi-conservative. Semi-conservative are changes that generally persist. That means they generally help in most cases, but there are some cases where they don't help. There are some cases where they're actually kind of lethal. And that might be, for instance, if you're an organism that lives in Yellowstone in the springs. I guess the springs are coming out like boiling hot. They're like super hot, but you still find life that grows in those ponds. They can tolerate those extreme temperatures. If they had some kind of a protein malfunction or a mutation or substitution where the proteins that keep them alive in spite of all that heat somehow can't tolerate that heat anymore, then the organism dies, and that protein doesn't survive to the next generation. And so maybe that organism would still survive outside of those what do they call them, the springs. There's a name for that. Is it Old Glory or something? There's a spring that I'm thinking about at Yellowstone, which is like extremely hot water coming up, but you still find life forms that live in the pool of this boiling water. It just cannot be killed because it's able to live in extreme conditions. It also lives in regular conditions, but the thing is, though, that if something happened to its proteins that keep it alive in that extreme condition, then it would die, and so we'd call that kind of a semi-conservative type of alteration. We have another alteration called non-conservative, which means it's lethal. It will kill the organism. If this thing happens, you're dead. And so therefore it's going to be, and I hate this term, edited out by evolutionary pressures. It sounds like some kind of a James Bond thing. Well, James, you're about to be edited out. It just basically means that the organism that has that gene does not survive to give that gene on to offspring. And so the gene stops there when the organism dies. So that's non-conservative. And so there's other things that are involved with those particular with these other particular definitions, but I hope that that's for this discussion that's sufficient. So getting back to these amino acids, we were talking about the physiochemical properties, and you can see that some of the amino acids, I mean, I'm not a chemist, but if you're an O-chemist, like an organic chemist, you should be able to look at some of these amino acids and kind of think, oh yeah, sure, I can see that this is polar, this is, where one side is attractive to different kinds of water molecules, whereas the other side may not be attracted to water molecules. Hydrophilic and hydrophobic. And so you have these different properties which are actually looming in each of these amino acids. This is what makes up your protein. So your hair, your skin, your hands, your bones, your eyes, everything that you are, and you are so much more. But everything that you are physically is built out of these kinds of bricks. So if this was if you were a giant Lego creation, these are the Lego blocks right here. So we have these components. These are the things that go into making proteins, and they have different kinds of properties. And we say that they're similar if, for instance, you're replacing one protein amino acid if it's polar with something else which is polar. We say that it's similar. It may not be the same amino acid, but according to its physiochemical property, polar and polar are similar. The molecule is different, but they behave similarly. They're also hydrophobic, hydrophilic. Some of them have different types of charges. They're positive charged, or they have negative charge, or they're basic or acidic. Here are just some sites online that you can go to if you'd like to learn more about types of properties. But there are, you're using these properties now to kind of determine how similar is one sequence of protein to another based upon, well, do I have the same number of polar amino acids in one sequence as I do in the other. And so you're really actually kind of doing this comparison using not just bases or not just amino acids and bases like we saw in DNA, but we're looking at a bigger picture. And it's for this reason that this becomes actually much more complicated when you're running things like BLAST. With DNA, you don't get this necessarily, but with protein, you can actually make some very sophisticated comparisons. So I think I've made this point before, that amino acids determine the shape and function of the protein. And so what we're doing is, if we know that the amino acids actually control the shape and function of the protein, we can look into certain pieces of knowledge about each of the amino acids in that protein to find out, well, what kind of hydrophobicity is there? What kind of, is it going to attract water, or is it going to repel water? And how does that property in this sequence, in the first sequence, compare with the other sequence? And so you can use that as a comparison metric. So you can, actually getting back to the question here, can we quantify sequences by physiochemical properties? A resounding yes, you can. And so we're not just looking now at just, if you go back to, let's see, here's your protein. We're not just looking at comparing strings now, but your comparison is actually getting a little bit more complicated at what is represented by each of these amino acids here. So here we go. If we're studying what's called these amino acid substitutions. That you have a first sequence amino acid over here, and you have a second sequence down here, and you find that there's something different about those two sequences. In other words, the sequence that I have is behaving somehow differently from another one, and so we're looking now to see that substitution, or that difference that makes one sequence different from the other. We're studying that particular area. But this is actually quite interesting, though, and that is that if you have a protein here which is just this LLE, in species A you have an organism which began with ALA, and species B you find that ALA has become LLE. You're looking to see how this substitution could have happened in terms of going from ALA, which was the first organism, to LLE in the second organism. And so you can look at different numbers of mutations. In this mutation, for instance, ALA, it just mutated directly into LLE with just one mutation. But by looking at, in fact, if I go back to this table, I don't mean to make anyone dizzy here, the Codon Table, which is right here, you can see that it's like that mathematical game, Send More Money, where you're trying to figure out, like, for each character it represents a number, and you're trying to figure out, like, how you can add letters together, meaning that each letter has a number, and have a problem that actually makes sense. But you're basically saying, how can I jump from where I am, LLE, to the next thing, to the next thing, to the next thing, using this table? And you can find that in this, it's like a game, I guess. In this scenario, you can actually jump from ALA to PRO, to SIR, to LLE in 3 mutations. And so you could say, even though I have the same protein here, could I argue that actually they are quite distantly related to each other, because this first protein has undergone 1 mutation, and the second mutation, or the second protein I'm looking at, has undergone 3 mutations to go from the DNA that it originally had to the protein which I know it to be now. And so this will give you some ideas about how different things are from each other. And so we call this the model of evolutionary change in proteins. And I'm going to gloss over the math here. Don't worry about that. But basically, you have this. You have a very simple logarithmic function, which actually comes from something called information theory, which is you're looking at the fraction of the probability of a mutation occurring in an amino acid. So that means that of all the things that I know about this amino acid, how many times have I seen it been replaced in nature when I compare from one organism to another? How many times have I seen this amino acid being replaced by something else? I mean, can it easily be mutated? And then I ask myself, like, well, what is the frequency of that particular amino acid in a large set of sequences? How many times have I seen it at all? And so I put this into this equation here. I attach the log function to it, and I multiply the whole thing by 10 to basically add it's normalizing in a sense. But I get a number that comes out, and that number gives me what's called the odds ratio. And that's how I can kind of tell how random was this change. This is like one of the tests you can use in your evolutionary change in proteins. If the thing is equal to 1, then you can say that it's likely the substitution of J, or substituting one amino acid for another, is no more likely than the chance of finding that first amino acid randomly in the protein. In other words, nothing big is going on. But if I find, for instance, that it's greater than one, the substitution is evolutionarily conserved. That means that it's doing something good for the organism. Or rather, I should say it's not killing the organism. So it's being conserved. It's always being passed on to the next generation. However, if I find that this number that I get out of this equation is less than 1, it's like a small number, like a 0 point something, then it's probably lethal. It's being selected against. That means it's not doing any favors to the organism. And so you have these like different types of checks and things you can use to see how whether these substitutions are actually making any difference. I'm going to start, I think I might stop here. I think we have a whole bunch more slides. We'll hit this on Monday. But I wanted to just to kind of leave you with one message here, and that is that when you see these kinds of alignments here, when you're actually working with proteins, what we're talking about now is that we're no longer just comparing one string with another. That happened before when we were working with DNA. But now we have evolved our thinking, and so that it's not just one string with another, but rather it's one string and all of these physiochemical properties and the likeliness that these changes could possibly happen. It's all of this other stuff which is being kind of combined into one number, if you will, one metric, and that metric is then multiplied across for all the different organisms or all the different amino acids, and that is the comparison basis. It is that type of comparison basis that we use when determining the E score. This is a question that I had that was just before the breakup of class, and so some of you may have left the room by that time. But let me just go back to the results we see here from BLAST. Here we go. So we have the E value right here. So this E value is using a similar theory, but it's applying it to DNA. It's not it's not as sensitive when you're working with proteins, but it's using that kind of probability theory to determine, was this random, really? Or was this actually forced by something? And so what could it possibly be that would create this change? Anyway, that's all I got for now. I think that I've kept you over a minute, and I'm sorry about that. When we come back, we will finish these slides, and I think that we're going to do some more analysis over our own proteins using some of this theory. All right, everybody, so take it easy. Go outside, get some sun. It is beautiful outside, just gorgeous.